Welcome, everyone. Today, we are going to explore one of the most fascinating creatures on our planet, the Sphenodon, commonly known as the Tuatara. Imagine walking down a modern city street and seeing a fully functional, steam-powered carriage from centuries ago. That is exactly what the Tuatara represents in the animal kingdom. It is a living time machine. By studying this incredible reptile, we can look back hundreds of millions of years and understand exactly how modern reptiles evolved from their ancient ancestors. Let us dive into its story. Now that we have introduced our subject, let us understand its place in the animal kingdom. Why do we call the Sphenodon a living time machine? This creature, which is found only in New Zealand, is the sole surviving member of an ancient reptilian order called Rhynchocephalia. While the modern lizards and snakes you see today belong to the order Squamata, the ancestors of the Tuatara branched off on their own completely separate evolutionary path over 200 million years ago. Because its physical anatomy has remained remarkably unchanged since the time of the earliest dinosaurs, biologists frequently refer to it as a living fossil. It is a direct breathing link to the Mesozoic era. Building on its status as a living fossil, let us look at the specific physical traits that make it so special. The Sphenodon retains several primitive anatomical features that have been lost or heavily modified in most modern reptiles. For instance, it has a classic diapsid skull, meaning it has two complete temporal openings on each side. It also has amphicoelous vertebrae, which are uniquely concave on both ends, and a highly developed parietal eye, often called a third eye, right on the top of its head. Understanding these specific ancient traits allows scientists to reconstruct the baseline anatomy of early reptiles before they diversified into the thousands of different species we see around the world today. Having discussed its primitive features, let us now visualize exactly what this ancient skull looks like. On your screen is a detailed diagram of the Sphenodon's diapsid skull. Notice the two distinct openings on the side of the skull. The upper one is the supratemporal fenestra, and the lower one is the infratemporal fenestra. These openings are framed by the upper and lower temporal arches. This double opening structure is the defining characteristic of a classic diapsid skull. Additionally, if you look at the very top of the skull, you can see the location of the parietal eye, a remarkable sensory organ. This diagram perfectly illustrates the foundational reptilian anatomy that the Tuatara has preserved for millions of years. Now that we understand its physical anatomy, we must ask, exactly how long ago did the Sphenodon separate from other reptiles? To answer this, evolutionary biologists use a brilliant mathematical concept known as the molecular clock. The core idea is that DNA mutates at a relatively steady rate over time. By comparing the DNA sequence of the Tuatara to the DNA of a modern lizard, scientists can literally count the number of genetic differences between them. Because we know how frequently these mutations occur, we can use those genetic differences to calculate exactly how much time has passed since these two distinct lineages shared a single common ancestor. This concept of counting mutations leads us directly to the mathematical formula for the molecular clock. The equation is T equals D divided by the quantity 2 times mu. Let us break down what each symbol means. The capital letter T represents the divergence time, which is the total number of years since the two species split. The capital letter D stands for the genetic distance, meaning the measured proportion of base pair differences in their DNA. The Greek letter mu represents the mutation rate, which is how fast mutations accumulate per million years. We divide by 2 because both the Sphenodon lineage and the lizard lineage have been mutating independently since they split. If the genetic distance d increases while the mutation rate stays the same, it physically means the species diverged a much longer time ago. Before we move on, let us see this formula in action with a concrete example. Suppose a biologist compares mitochondrial DNA between a modern tuatara and an iguana. We are given a genetic distance of 0.40, meaning a 40% difference in base pairs. The known mutation rate is 0.0008 mutations per million years. We want to find the divergence time. First, we substitute our given values into the molecular clock equation. Next, we multiply the mutation rate by 2 in the denominator, giving us a combined divergence rate of 0.0016. Finally, dividing 0.40 by 0.0016 gives us 250. This final answer physically proves that the Sphenodon diverged 250 million years ago during the early Mesozoic era, perfectly matching the fossil record. So, we have proven that the Sphenodon is incredibly old genetically. This brings us to another fascinating question. How fast has its physical body changed over that massive time span? While the DNA of the Sphenodon has steadily mutated over 250 million years, its physical skeleton has remained remarkably similar to its ancient ancestors. To quantify this, evolutionary biologists measure the rate of physical change using a specific mathematical unit called the Darwin. By comparing the size of a specific physical trait in a fossilized ancestor to the size of that exact same trait in a modern Sphenodon, we can calculate a standardized metric. This allows us to prove mathematically just how slowly the physical form of the tuatara has evolved. To actually calculate this rate of physical change, we use Haldane's formula for evolutionary rates. The formula is r equals the natural logarithm of x2 minus the natural logarithm of x1, all divided by delta t. Let us define these variables. The lowercase r is the evolutionary rate, measured in Darwin's. x2 represents the modern trait size, while x1 represents the ancestral trait size. Delta t is the time interval between them, measured in millions of years. We use the natural logarithm, denoted by ln, because it measures proportional change rather than absolute change. This is crucial because it makes it mathematically fair to compare the evolutionary rates of very small animals with very large animals. If the difference between the trait sizes increases, the evolutionary rate r will also increase. Now that we have our formula, let us apply it to the Sphenodon's lower jaw. We are given an ancestral jaw length of 40 millimeters from a fossil dated 200 million years ago. The modern jaw length is 42 millimeters. We need to find the evolutionary rate in Darwin's. First, we substitute 42 for x2, 40 for x1, and 200 for delta t. Next, we calculate the natural logarithms, giving us approximately 3.737 minus 3.688. Subtracting these gives a tiny numerator of 0.049, which represents the total proportional change. Finally, dividing by 200 yields an evolutionary rate of 0.000245. This incredibly small number physically proves that the tuatara's anatomy has barely changed at all. This mathematical result we just calculated leads us to a profound realization. The evolutionary rate of 0.000245 Darwins is exceptionally close to zero. What does this mean in the grand scheme of biology? It mathematically proves that the physical anatomy of the Sphenodon has remained almost entirely static over hundreds of millions of years. To put this into perspective, mammals often exhibit evolutionary rates of 1.0 Darwins, or even higher, meaning they change rapidly over time. This stark mathematical contrast highlights exactly why the tuatara is uniquely significant. It is not just an old animal, it is a perfectly preserved biological window into the ancient past, surviving global mass extinctions while retaining its original form. We have covered a lot of ground today, so let us summarize the key takeaways. First, we learned that the Sphenodon is the sole surviving member of the ancient order Rhynchocephalia, making it a true living fossil. Second, it retains primitive anatomical features, such as a complete diapsid skull and a parietal eye, providing an irreplaceable model of early reptilian anatomy. Third, by using the molecular clock, we mathematically verified that its lineage diverged over 250 million years ago. Finally, through Haldane's formula, we proved that its physical evolution has been incredibly slow compared to other vertebrates. You now have a comprehensive understanding of why the tuatara is a priceless biological time capsule. Great job following along with the math and the biology today.